Okay, thank you. So I'm Fernanda Cerqueira, scientist at the Fox Foundation, and first I would like to thank Viv, Shalini, and the organizers for this great opportunity. Today, I'll be discussing biomarkers for GBA1 targeted therapies in Parkinson's disease. Before getting to the biomarkers, I would like to give a high-level overview of MGFF strategic research agenda, give a high-level overview of the clinical pipeline in PD and associated markers, and then let's go to the gaps in the space and opportunities for collaboration. So our research agenda, our mission, our vision is a world without Parkinson's disease, and our mission is to go out of business. And that's literally, you can find that in our website. To fulfill our mission, we fund aggressively several programs which all converge to the same goal, which is enable the development of new therapies to slow disease progression or to ameliorate symptoms in symptomatic individuals or to identify individuals at risk and come up with strategies so the symptoms, to prevent symptoms onset. Our strategic research agenda is split into 4 main areas. Our first area is a better understanding of the disease biology of PD's heterogeneity. What is the biological definition of PD? Our second area is we have programs which aim to de-risk novel targets while also funding well-known targets linked to PD, and we try to push them to early stages of clinical development. One example is GBA1. Our third area is programs which can bring the community together. We have programs to educate patients and the scientific community. We bring stakeholders together and just get different opinions in the room and move the field forward faster. And our fourth area that became neon there, I don't know why, is the goal of our talk today is the development, the discovery of novel biomarkers and the development of tools to measure those biomarkers in a robust, quantitative manner. So let's start with the clinical pipeline, which was already covered yesterday by Simon, but I'll just give our flavor how we are viewing the current pipeline. You can scan the QR code that's going to take you to the pipeline, which was spearheaded by our chief scientist, Brian Fisk. So there are many approved drugs for Parkinson's disease symptoms, actually since the 60s. Most approved drugs, they address motor symptoms, and the main target is dopamine. We still don't have to date a drug approved to slow disease progression. However, now the landscape is changing. The current clinical programs in PD are 50% dominated by programs aiming to slow disease progression. And we can see here that there are many pathways represented in this space. The endolysosomal pathway is quite represented with LRRK2, GBA1, and autophagy programs. There are, to date, and I know that because of Simon's talk, 9 clinical programs in Parkinson's disease, in GBA for Parkinson's disease. Most GBA1 programs are in phase 1 and 2, and yesterday someone asked a pretty important question. Why are biomarkers needed if we are mainly following clinical outcomes? Each stage of clinical development is dedicated to answer very specific questions, and biomarkers are critical to answer most of those questions. In the phase 1 study, you have to assess toxicity, tolerability, and target engagement. I think we've heard a lot of target engagement talks. Only a biomarker, in most cases, a biomarker can give you a measure of target engagement. In phase 2, you have to prove a mechanism, proof of biology, and biomarkers can help you to understand if a pathway, a given pathway was modulated, and it can even help to explain your clinical results. If you had a good clinical result, an expected clinical result, or the lack of a clinical result. And phase 2 is also very important for dose optimization. And then in phase 3, the biomarker questions are very different. It's not related to the target or to the pathway, but when companies want to ask questions such as, which patients I should enroll, which are likely to respond to this therapy, a patient enrichment marker, or which marker I can use to monitor the disease progression. Or even more exciting and harder to get, is there a marker which can predict a clinical benefit? That is and the regulatory requirements for those markers with those contexts of use that I just mentioned in phase 3, they have very high requirements. The FDA has a very high bar to allow a marker and an assay to be accepted for those types of markers. What are the GBA1 programs and associated biomarkers? This is a very nice framework that Pablo Sardi put together, and he presented last year in our endolysosoma workshop, which we hosted at the Michael J. Fox Foundation. So the patient selection, it's mainly used the GBA1 status, as we have heard many times today and yesterday. The levels of the drug in the CSF and plasma is another very important measure. The target engagement, I think we all know by now that the enzymatic activity of GCase or the accumulation of substrates are the main markers for GBA1 asset. And then we have the pathway modulation, which is how the target engagement modulates the surroundings, the adjacent pathway, and how that can modulate the cellular biology until changes the disease trajectory. And then how do you follow that? By the disease markers, the alpha-synuclein aggregation, the NFL. That's the hope, to get to the point where you are actually modulating the disease trajectory, and you can track that through those disease markers. For the purposes of the talk today, I'll mainly focus on the target engagement and pathway engagement because they are very related to the GBA itself. The disease biology, we can claim, they'll be useful for 2 trials, for many other trials. So another nice table from Pablo Sardi. We don't have all programs here, but I like this table because it captures the diversity of mechanism of action of the programs. We have allosteric modulators, we have chaperones, we have enzyme replacement therapy, gene therapy, and venglustat is here, even though it did not show efficacy in Parkinson's disease. Venglustat, I think it has a very unique mechanism, which is reduce the substrate accumulation. And venglustat showed from the get-go, from the very beginning, years ago, how tracking a substrate reduction was enabling the program to make decisions and to move to the next phase. So not only venglustat was important in this regard, you see that the other companies and programs which came later are following similar markers of substrate accumulation and GCase activity. The company, all in the right side of the table, we can see the main markers being followed by the programs and what's being the reported alterations in those markers. And the programs are showing alterations in glucosylceramide in the CSF, in glucosylceramide in the plasma, in GCase activity. In some cases, they show alteration in both markers. Now I will get a bit technical and I need you to bear with me because this is important. We are going to talk about the gaps in the space. There are many different ways to measure GCase enzymatic activity. There is cell-based, fluid-based, there are the substrate for those assays. They are synthetic and bound to a fluorescent probe, or they are heavy labeled lipids. And to add more complexity, there are many assay variables. pH, there is the use or not of CBE. There are the quantification is mainly relative. It's not arbitrary values for fluorescence studies. So we have so many variables and so many different types of assays, it's almost nearly impossible to compare results from one company to the other. I don't know any company in the space, and correct me here, that are using the same methodology to measure GKs. And a 50% activation in one program using one assay could very well mean 20% activation in another program using a completely different assay. So that's okay because the companies are comparing baseline with treatment, following the same individual before and after the dosing. So in the boundaries of the program, the assays are fine. The assays are still enabling decision-making. However, for the scientific community, when we look at the data, we don't know how to interpret. We don't know what the threshold, how much GK's activity a good program should be aiming for. On the other side, let's talk about substrate. Substrate accumulation is cleaner. We don't have so many variables as the GKS1 assay. However, those species, glucosylceramide, especially glucosylsphingosine, they are very hard to measure. They require specialized methodologies. Your regular lipidomics is not going to detect those species. And they are the aspect of subspecies. There is no such a thing as one glucosylceramide. That was emphasized yesterday on the opening keynote talk. We have many subspecies of glucosylceramide with different carbon chain lengths and so on. Glucosylsphingosine, we are lucky we have only one, but they are still very hard to detect, very complex metabolism. This is one example to make my point that I have here 3 publications from 3 different groups using different methodologies and different matrices, and they are displaying very different results between PD, idiopathic PD and GBA1-PD. It varies from 50% of changes in GK's activity to 10%. Of course, someone can ask, well, but it depends on the variants in the study. But even if you look at the first 2 studies and you look at the same exact variant, there is a big difference of GK's activity being reported. And of course, it can also, we can also explore the matrices of choice. Maybe a monocyte assay is much more sensitive than a dry blood spot. Yes, however, the matrix of choice is also part of the assay development. It's part of what is driving the decisions in the company's programs. So moving from GKs to substrate accumulation, glucosylceramide has been reported to be increased in plasma and CSF of GBA1 carriers according to literature, and it has also shown to be reduced by several programs targeting GKs. However, while most programs are reporting total glucosylceramide, most differences are observed in certain subspecies. For glucosylsphingosine, we only have reported differences in plasma and Dan showed that very nicely yesterday. In CSF, we have not seen published data showing differences in CSF between GBA1 carriers and idiopathic PD. However, until very recently, because Denali and Dr. Gillian Klaus is here representing and team, they have shown a fantastic PPMI data where they saw a 40 to 50% increase in glucosylsphingosine in the CSF in GBA1 carriers compared to the non-carriers. We got very excited about that result because you can follow that toxic species on a matrix of interest. The only caveat is, unfortunately, the quantification, the absolute values, the picogram per ml, did not exactly match what has been published previously. And I think that we had a very good discussion about that yesterday. That triggered round robin where we sent samples to different laboratories and CROs, identical samples, and we retrieved very different results. Here we sent samples to Lipotype, Nexia, Merck, and Denali, very robust quantitative platforms, and the results varied from 0.22 to 5.8 picogram per ml. That just shows that even the best laboratories in the world still did not get a hold of the glucosilsphingosine quantification in the CSF. The pathway modulation markers are still highly exploratory. The companies are pursuing metabolomics, lipidomics, proteomics, Olink, but we still don't understand how the changes that are being reported translate to a lysosomal benefit. And I think Dr. Susan Pfeffer asked a very good question yesterday. What does it mean seeing cathepsin B going up? Or we don't know yet. We need more natural history data. We need to see how that pathway is dysregulated in GBA1-PD and then try to revert back to normality. So summary, we have many open questions and opportunities for collaboration here. We know there are gaps in the GBA1 measurements. We need to harmonize the assay. Our goal, our wish, is to have a gold standard that everybody could use and we could establish a clear threshold. We are aiming for X percentage of increase in GBA using that specific gold standard, that's this specific assay. And if we cannot develop that in the CSF, let's then establish a correlation between peripheral markers and CSF modulation. Without that correlation, I think it's very weak to base clinical development only in peripheral markers. We have to fix the glucosilsphingosine, and we are open to recommendations of other CROs or who can help us to solve that issue. Pathway modulation, we still need highly sensitive panels. We need to understand how the pathway modulation is linked to an improvement in the lysosomal overall health. With that, I'm just going to mention that we are tackling some of those gaps through funding studies that we are currently funding. We are funding a round-robin study for Roy Alkali, thanks to Dr. Peter Bauer from Centogene, who provided samples of GBA1 carriers of multiple severities, and we are comparing 3 platforms for GBA1 activity. Hopefully, we'll be able to make a recommendation at the end of the study. I've mentioned the glucosilsphingosine round-robin already, and for the lysosomal pathway, we have several activities, including the GBA1 consortium that was mentioned by Zev. We have a fatty acid profiling, which Dr. Sarana Fanning is investing, and she has seen wonderful differences in GBA1 carriers and other studies. Before my time runs out, I would like to emphasize this very large PPMI study that we started at the end of last year. We are going to generate robust natural history data across our PPMI cohort, looking at several pathways of interest, including the lysosomal pathway. We are also looking at the disease markers. It's a longitudinal analysis, so we will have potential markers of progression. These are the opportunities for collaboration I already emphasized. Let's standardize the GBA assay, please. Let's improve glucosilsphingosine measurements. And if someone has a very good mechanistic assay, which could be converted into a biomarker assay, let's have a conversation. I just would like to quickly thank all my colleagues, especially Dr. Shalini Padmanaban, who is investing in all those efforts in our leadership level and who is also a great mentor. My colleague, Audrey Dinman, who brings all the right stakeholders together to allow us to move forward. The GCAN meeting organizers, our wonderful advisors, and all the companies who are helping us to identify those gaps and what is important to move the field forward. Thank you very much.